Vineyard today. Oh, great. Thank you soon. Welcome everybody to the CCV Skills Seminar. Today we have Michael Vineyard, who is a PhD student at Harvard University. He works on developing methods for the analysis of single cell genomics data and also works with CRISPR-Cas9 based genome editing tools, for example, base editing, to study specific cell states during cancer progression. He is funded by an NIH award through the NCI, and he focuses on applying deep learning frameworks to single cell molecular data. So yeah, Michael, he's a PhD student also in the cancer program at the Broad Institute. So doing lots of exciting work in cancer and single cell that we'll get to hear maybe a little bit about today, but otherwise he's going to teach us a bit about single cell dynamics. So I would tell you to clap, but it's kind of weird on Zoom. So just join me in welcoming Mike. And yeah, take it away.
Thanks, Monica. And yeah, it was really great to hear from you when we were talking about, when you told me about this seminar. And yeah, I have to say you were probably one of the first friendly faces I knew in the Gets Lab when I joined several years ago. So yeah, this is super fun for me. And I really like talking about these things in general. And yeah, I found it was really like a pleasure to put this together. Hopefully it goes well without too many technical snags. I was realizing that, as I told you before, there are a lot of intricacies when it comes to trying to go between collab and slides and everything. So hopefully we can get it all tied together, but just anybody, please feel free to interrupt me if you have a question. Correct me if I'm wrong, but this goes for 1 hour, I believe. So that's roughly what I've prepared for, till I suppose, so what would it be, 1:30, right? Okay, so that's what I'll plan for. And hopefully we can get through most, if not all of it. And then everything else, especially if I find any holes, I'll try to put these online afterwards to go with the website. But basically, a general outline of what I was hoping to accomplish today. As you saw from the title, we'll be talking about cell dynamics from single cell measurements. And first, I think it'd be nice just to get some footing in what the state of the field is. I'm not sure what the composition of people in the department are like here. So maybe, I would imagine that it probably spans, several different types of genetics, genomics, other sort of biology. So probably not everybody is super involved in single cell. And that's also, I feel like philosophically fine, because probably some of the problems we'll talk about today, I think, can be generalized to other things, even if single cell may not forever be like a popular measurement choice. So I feel like that's kind of something to consider when thinking about these types of assays and where some of the philosophies came from for how we analyze certain types of data. And so the second thing I think that would be nice is let's see, there's a question in the chat here.
Oh, that was just me. Yeah. And yeah, the collab notebook and everything will be available after the talk.
Oh yeah, I should mention this for those that came in late. Basically, what I'm going to do today and what I'm kind of starting to tell you now is I have a few slides prepared just to talk about the conceptual framework of what we're talking about today for cell dynamics. And then I have a tutorial where we can dig in a little bit to how a cell dynamics model, like one example of many, might work. And one thing I want to say just up front, I'll say it again later, is that there are so many different methods for cell dynamics and so many different approaches that it's really my goal also not to, it's really hard actually I realized in putting this together that it's really hard to even figure out if you're missing any, because there's so many different approaches that make different assumptions and consider different scenarios that if I left anything out, if you happen to be involved in cell dynamics and your tool or your favorite tool didn't get mentioned, I hope not to offend anyone. I really wanted to give a brief overview, and then I'll also provide some disclaimers eventually with where I'm definitely not an expert, and try to point to those who are experts and might be better suited for a tutorial in that area. So with that, the second goal will be to get familiar with a few of these tools for cell dynamics analyses. And the third goal is finally, as I mentioned, we'll kind of transition to this Google Colab type of environment, and hopefully we can generate a simulation learn cell dynamics. Initially, I had planned for an animation, but I actually didn't get the code. There was a few snags in going from just a normal Linux environment to Colab that prevented me from moving all my animation code over. So we have a static recreation of our cell dynamics today. We'll still get to see a whole path plotted out, but in the future, I can, as an addendum to this talk, I can add the animation code, and hopefully you can give that a shot. So first off, I'll just start super simply and, discuss what do we even mean when we say cell dynamics? And this is a really vague term. And even once you define it, it's still kind of vague. So in general, most people are familiar with this idea that single cell measurements kind of gave us this, they're readily appreciated for their ability to give us some insights or some picture of the heterogeneity within tissues that would otherwise be averaged over when we measure them using methods like RNA-seq or ATAC-seq, or this is expanding all the time, so like ChIP-seq even, and things like this. So, but the main idea that I think is kind of beautiful once you realize it is that the limit that you go down to is really single cell. So even though most times people are looking at kind of differences between clusters and differences in doing differential expression and things like this between groups of cells, whenever you're looking at a biological process that where the cells exist or kind of coexist alongside their progenitors. So for example, like a really common vehicle for this type of discussion is hematopoiesis, where your kind of like hematopoietic stem cells are coexisting with your more mature cells like plasma cells, neutrophils, et cetera, in this bone marrow environment. And so when you have all of these, and it became a really popular topic, probably in around 2016 to once these measurements kind of took off at scale to perform some sort of like dimension reduction and what's kind of commonly referred to as trajectory inference to map these cells to some process. And I'll talk more about these ideas later, but the key idea here is that if you are observing such a process and you sample your cells densely enough, you can actually kind of recreate the whole process. Of course, that's oversimplifying the task, and there's a lot that goes into doing that. And I think the verdict is still out sometimes for how we do that. But as I kind of already threw in a few jargon terms, I want to try to not use jargon where possible, but part of today's talk will also be introducing you to some jargon that's kind of necessary when surveying this area. And so the first term, is, and I won't go through each one of these, but in the written document that I have to go along with this that I'll send to Monica after for upload to the website, I'll include some definitions and pointers to articles where there's good explanations for each of these things. And if you do have a question, or if I kind of like skip through something that is useful to define, or you'd like me to go back to, please just let me know. So I don't know if this will play, but, oh yeah, and there's kind of some music to go with it, I guess. But, when we are thinking about cell dynamics and what this video is trying to capture is, this is a live cell imaging experiment, which I actually realized, I mean, I'm not at all in the imaging field, but I realized when I was searching for this, that trying to find such cool videos is actually really hard when we're trying to figure out, what real cell dynamics looks like. And so this is some neuronal stem cell differentiation process that I'm not super familiar with, but the general gist of the video seemed relevant. And the reason I'm showing it is because most of the times when we build models and we're building analysis methods for biology, what we're really trying to do is recreate some meaningful representation of what happens in real life. And so the reason I show this first is because, if we could, we would get back to something that is quite realistic. The second thing I've put up here is this Conrad Waddington's epigenetic landscape. And this is an idea that, it's worth considering how, and you probably hear this in a lot of papers, it's almost kind of faux pas, I feel like, to put this on slides if you're giving a research talk, not to discourage anybody from doing that, but I feel like we see it a lot. So the kind of excitement around using this in modern biological methods is that there's this idea of a driving potential where if you have some stem cell state, so that would be this marble at the top, it's going to roll down some hill of a predefined trajectory or landscape, whatever you want to call it, into some more defined cell state that's also considered to be more stable. So there's a couple of ideas to go along with this, where one would be I don't really know how to call it, but the axis that's kind of going backwards or, forwards towards the page might be like a driving force like time or some other biological driving force, and the z-axis here going up and down, or the y-axis, I suppose, going up and down would be referred to as like a potential, where you can think of this like Newtonian physics, where potentials that are high kind of have this, not yet low kinetic energy, and they have a high potential to move into some other space. And then the x-axis, of course, might be something like a gene expression or some other measurement that you can quantify to describe a cell. And the underpinnings of this might be something that's like a programming, like transcription factor programming. And it's kind of worth thinking about that this was drawn before we even really had, we hadn't even solved like a crystal structure of DNA yet. So, this kind of really predates like our modern conception of genetics and epigenomics and genomics. But it was actually really insightful for a start to thinking about dynamical systems. And actually, I didn't mean for this to get sent behind, but the last thing I included here is a representation from a more recent paper called Waddington OT, which is published from the Broad. And this is kind of using these fundamental principles of potential in cell dynamics theory to measure multi-time point single-cell RNA-seq data. So I'll talk a little bit about more things like this shortly. So going back to our idea of this like heterogeneous sample that gets split into these different subpopulations, for the purpose of today's discussion, really, I think I would encourage you to think about a cell as something that can be looked at like a vector. So this might be obvious to some of you. For others, maybe it's a little bit more foreign, but we can really think about each individual cell like a vector of 20,000 or whatever have you number of features. And to that end, we can think of each cell observation. So like each cell you might get from a single cell experiment, like a snapshot of that cell at some point in time. And so, as I mentioned before, there are methods to perform dimension reduction and align cells with respect to time along some biological axis. And when we have this continuum, and if we wanted to learn the dynamics associated with this continuum, we could describe the gene regulatory network as something like F, where F is a function of the current gene expression state that controls or dictates where you'll go in some future state that controls your kind of path forward. And so there's a lot of options to analyze dynamics. Most of these, however, kind of fit neatly into this idea of trajectory inference, where they don't really offer much underlying insight into gene regulatory programs. So we've gotten really good as a field at doing dimension reduction and doing pseudotime alignment. And so pseudotime in this case kind of being your time along this progression along a biological axis. And one of the main challenges associated with this is that, for the most part, aside from a recent Nature paper, these assays are without resampling. So they're destructive. You see a cell once as a snapshot, and then you never see it again. And you have to rely on the population level differences of cells to understand where that cell fits within its environment. The second, more complicated part, and these are concepts really borrowed from Alan Klein at HMS. He kind of has this really nice paper with a postdoc who will look at some of their work today, but his name's Caleb Weinrib. They have a series of papers where they outline kind of the limits of single cell analysis and what you can do with various clonal mechanisms and lineage tracing and things like this. So the key challenge that they kind of hunt after in multiple papers is trying to understand how cells can be viewed as like intermediates or current snapshots of some mechanism and process. And the key complicator to this is that you can have multiple mechanisms that kind of conflate the state of a cell and make a cell look, you can have two cells that are identical in terms of their gene expression state, but they're in the middle of two different processes. Fortunately, we typically tend to know something about the biology of the cells we're measuring, and this can help us constrain our analysis methods. So, okay, I'm just checking the clock to make sure I leave enough time. So this is the area where I have to provide a disclaimer, because I'm truly not an expert when it comes to RNA velocity. And most of the dynamics work I've done has been kind of on the other branch of cell dynamics. And I'll explain that a little bit more in the next couple slides. But I just wanted to give a brief overview just so that we at least have it out there, and kind of considering this as like a survey or an introductory survey of cell dynamics. So there's this paper from Joella Lamano in 2018, and they're really borrowing from these concepts that had kind of been around from bulk sequencing, where in bulk RNA sequencing, we could use kind of tags or specific experimental designs to tease apart which reads were spliced and which reads were unspliced. And the reason people were interested in doing this is because transcriptional splicing is a constant process, more or less. And biology requires cells to, if you want to change cell states, it's our kind of general accepted understanding that we are required to change the expression level of gene programs in order to do this. And so what really being able to quantify spliced and unspliced reads gives us is that since the splicing process is constant, we can start to see what the future of cell states might be. And to explain this a little bit more graphically, we have some constant rate of transcription alpha for some gene, and this is creating some unspliced mRNA. And as the splicing process, so beta, this is constant, so this is defined as 1, is going along, it's creating these spliced processed mRNAs, which are eventually degraded. And so when we count or sample mRNA molecules from a cell, we kind of expect these processes to look like this, where, if the transcription is on, and of course it's probably never this clean, but if you have some transcriptional burst that causes transcription to happen, you'll have that at some rate alpha, and it will eventually turn off again, or maybe not, who knows. And during this process, you'll have some accumulation of unspliced reads, which, this relative proportion of this will go up with respect to time. And then as it goes down, it will decay. And then, of course, for degradation, this kind of follows this at some relative rate. And so when we see some abundance of unspliced reads gathering for a given gene, we can kind of infer that the splicing, so hasn't yet processed these reads into spliced reads. And since we are seeing a buildup of it relative to the amount of spliced reads we have, we can infer that there's some kind of like increase that wasn't previously there, meaning that if at some steady state, along this gray dotted line, you would kind of expect, if there was no change in the biological system, you would expect the ratio of spliced reads to unspliced reads to stay the same. And of course, this is somewhat of an oversimplified model, but this is kind of a nice generalization for thinking about how cells change. And the reason that this paper was such a kind of like, I'm not really sure the word, but like such a big hit, so to say, was because people had already generated by 2018 a plethora of single cell data. And so this was something that people could go back and look at data we already had and kind of remine it to find out additional information about their cell systems. And I think probably when it comes to methods development, those are probably some of the most powerful paradigm shifting, so to say, methods that are invented or will be invented in the future, is anything where we can take, something for cheap. So to look at, to get new results from data we've already generated is usually pretty exciting. So as I mentioned, there's a ton of software tools for this, for both spliced and unspliced reads. as well as systems that don't use this. So Velocito and SCVelo are kind of like staples of the field. And I know Adam is a co-organizer of this talk, but he's instrumental in making some of these tools available to the public, especially when it comes to VAE-based tools. And they have a recent tool called VeloV. I haven't had the chance to use it yet, but I love all of their other tools, so I'm sure it's great. And there's probably more tools I'm leaving off of here that I haven't really thought about or had the chance to work with yet. But I'm predominantly a Python programmer, so that's most of my exposure. And I think most of the development in this field has kind of centered around Python, at least for dynamics. Of course, there's other tools written in R for single cell analysis at large, like Seurat and so on. So the software tools that are, there's kind of this core group of tools for working with splice and unspliced reads, where Velocito is this kind of preprocessing tool that you can then say, okay, I've now quantified my splice and unspliced reads. Now how can I make sense of them and turn to some of these other models? The software tools for modeling cells as particles. So I haven't really talked about this as much, but as I mentioned, Alan Klein's lab, who have these kind of nice series of philosophies when it comes to working with dynamical systems and single cell data. They have a nice method called PBA. I already mentioned Waddington OT. The method we'll actually go through in the tutorial is called Prescient. But there's also several other, kind of similar, in some sense, methods where they're really trying to frame this as a dynamical systems problem. Cell rank, I think, is quite a popular tool in my experience. Actually, it's one of the easiest to work with. We'll check out one of their what they call kernels today to get a pseudotime, and there's several others. So this is by no means an inclusive list. Okay, so I might have this might feel a little bit out of order, but I just briefly wanted to touch on this idea of thinking of cells, thinking of single cell measurements in the framework of a dynamical systems model. When single cell analyses really started to take off, people kind of got excited about this idea that, we have all of the genes measured in all of the cells, and, this is such a high resolution measurement. We're finally going to be able to solve gene regulatory networks which otherwise took, decades or, are never solved as of today, to work out even, quote, simple gene regulatory networks. And the reason I say simple is because to really constrain your assumptions to something that you can computationally work with, you have to oftentimes work with, only a handful of genes, from 5, 6 or so. The largest, I searched this a year or two ago, the largest kind of like high confidence empirically derived gene regulatory network I found was, around 19 or 20 genes. And so, yeah, this is a really big problem, a really hard problem. If you look now, even there aren't really good methods to do gene regulatory analysis in a dynamic sense, especially for single cell. And so that's not to say that the existing methods don't have good insights. It's more so to say, I think the field at large generally recognizes that this is an unsolved problem. And so, one way we might frame this problem, if we had never really thought about it before, would be to say, okay, for each gene, I'm going to simply write down a differential equation that describes the change in that gene expression level with respect to all the other genes in the system. And, we can sum this up to be one big gene regulatory network, f of x. And, this quickly, as you may guess, becomes kind of an untractable problem. So we can then start to think about this in a way that's a little bit more top down, so to say, where we can look at like overall attributes of a cell and try to make some kind of constraining decisions about what we think that cell is and where it should go within our dynamical system. And then we can look deeper at the gene expression levels. Excuse me. And so with that, there's this, I mentioned this before, this idea of potential where, as a cell moves through its pseudotemporal kind of space and maybe starts around some cell birth process and ends in death, that it might go through some kind of semi-stable states where these might be observed cell types or things that we can actually kind of measure in bulk. And of course, between those states, they might occupy very transiently some intermediate states, which are maybe a little bit harder to capture without such technology. And so the proposed framework for working with these is kind of framing these as a drift diffusion equation, where you can think of this like kind of a generic partial differential equation to describe some deterministic movement. So this would be, and I'll explain this a little bit more here, this would be movement that you kind of stereotypically see in systems. So, cell differentiation that leads to cell death, and kind of these pre-programmed things we think about when it comes to like really well-defined cell types. And the second term of this would be something like diffusion, where it's a little bit more spurious, a little bit more stochastic. And this is kind of the real life element that we think of when we think of biology and how biology can be messy. And I think it's kind of a really good question still is what contributions to cell fate and cell decision-making does diffusion make in a very general sense? So this is like an open question. And I think that, several people have tried to tackle this. The method I mentioned before, PBA, tackles this with building a KNN graph and then creating a Markov transition matrix between each cell state. And this is kind of like the kicking off point of a lot of the work I've done in my PhD, where we're trying to really drill down on what does this diffusion comprise of. So they outline this drift diffusion equation framework, but they basically say, we get kind of stuck when we try to solve this because it's computationally, intractable. It's a partial differential equation in a high dimensional space. So we don't really have a good way to solve these types of equations. And so in their case, they're able to take advantage and solve these Markov transitions by using a nice trick from graph theory, where you can do a very simple mathematical operation. And for some of you, this will sound kind of silly, but for others, maybe the terminology is useful as the Laplace transform on the KNN graph. And so they're kind of able to get away with this computational trick, but they say, this is by no means a definitive solution. And we haven't really parameterized at all this concept of G. And so this other model, which we'll talk about, and we'll start to transition to the tutorial called Prescient, they work within this framework to kind of tackle this problem where they say, okay, if we have some, and I apologize, I didn't mean to skip fast. If we have some observed cell state X at some current time, and then some future cell state that we might be interested in, or we want to predict what that future cell state at some time plus some delta time, we can try to model that as a neural network and learn the function that dictates the transformation from one state to another state with respect to time. And they can make this a generative process by adding in some Gaussian sampled Brownian noise. And so this would be, in the framework of our f of x plus g of x equals some movement, this would be their g of x term, and this would be their f of x term. And so I'll kind of just skip past. This is repurposed a little bit from a different talk. I didn't mean to include all of that. But the top-down kind of approach here, or actually, sorry, let me back up. We kind of come to a fork in the road, so to say, when we're modeling cell dynamics. I listed all these softwares, all these different methods and kind of philosophical approaches you can take, where you're looking at cells as either particles in a system, or you're looking at their individual components and asking, what does this say about the cell as it in a global sense with respect to its dynamics? And there's kind of two camps you can go into here. One might be the top-down cell information theory, where the cell dynamics are kind of inferred from the group. So this, you could imagine kind of building a nearest neighbors graph and comparing cells across their nearest neighbors. And the contrasting approach to this would be information that is, as I mentioned, kind of similar to like RNA splicing information. We can measure this in a cell without necessarily looking around at the other cells. And so we can get some information content about that cell. One really convenient method, which we'll look at in the tutorial, is called cytotrace, where they are essentially looking at gene entropy. And what that means is the basic kind of observation throughout biology is that more differentiated states tend to have fewer genes expressed because they've kind of become specialized and they don't need the flexibility or the plasticity to have several different gene programs active at once. And so they kind of ran with this general observation and added a few fancy modeling tricks to come up with this idea of pseudotime derived from just counting the number of genes expressed in each cell. The reality is that most methods take some combination of the top-down information and the bottom-up information to really model cell pseudotime, cell ordering, and cell dynamics. So this was kind of a whirlwind. I realized, like, as I was talking and as I was putting this together, even that this is like pretty hard to cover in just like a short amount of time. So if you have any questions, like, please let me know. Just really briefly, I'll show you a little bit. So the data set we'll work with today is this lineage traced single cell data set, meaning, you know, it's kind of split into, and this is repurposed from another slide, so you can ignore the train and test parts here, but you know, it's split into multiple experiments where you actually have these lentiviral barcodes that go along with each cell. And what that really enables us to do is grab the relationships between daughter cells and their progenitor cells at each time point. And so we'll look at this data set a little bit during the tutorial. And just as kind of like a motivator, from the models that we'll look at today, the general class of models, these are the types of simulations that you can get out of the models once you learn them. So what I'm showing you here is actually from my own research, but the model we'll look at today is kind of fundamental for how we put these general class of models together. And so with that, I will transition to the Colab notebook, but I also, as I'm kind of getting set up with that, I'll, you know, feel free to ask any questions or, you know, interject with anything that might be confusing. So, okay, so I just have to restart my runtime, it looks like. So if anybody's having trouble with the Colab notebook, let me know. Otherwise, I'll run through it and then we can address problems after. So I might have to just refresh my page here. Okay. Okay, so I created a repository to go along with this talk where I just put some code that would be helpful not to have to look at so that we can focus on the parts that we're interested in. And really, like, what I was hoping to do here is show you like the very base level of a really simple model formulation within this kind of published framework called Prescient. And I included some links below. We'll also grab, as I mentioned, the cytotrace pseudotime from CellRank, where they're using these other published, they have this really nice formulation of kernels where they're able to kind of take the best of every different world. And, you know, they're able to do dynamics with both splice and unspliced reads, as well as this more kind of top-down approach, as I mentioned. So hopefully this will work for people. Let me see, I think I probably, oh, here, I can provide the link once again. Okay, hopefully that works for you. And to get started, I just included here a link to clone the repo. And then if you already have it cloned, this was more for me while I was working on it to just pull the most recent one. Oh, whoops, I probably shouldn't delete the syntactic sugar that lets us do that. Okay, cool. So at least I already have it. Hopefully we won't have to do too many updates here. And then this should automatically detect the device you're using. I was able to get connected to a GPU by changing the runtime type. And I actually got that from a previous tutorial from last year. So that was super useful for me. And let me know if, you know, there's any adjustments I can make with the screen to make this more viewable. But to get started, I just have this function which will download our ANData object. For those of you that are kind of familiar with the scverse and scanpy and ANData, hopefully this will be really native to you. The other thing we're downloading is a checkpoint for a PyTorch model from Prescient's website. So I'll include the links to those at the bottom. So instead, since we don't really necessarily have time to train a model, even though, you know, their model is actually quite fast, it only takes maybe like a half an hour to train on a GPU. We'll skip that part today and we'll just start right from a trained model, but we'll still put together the models so that we can see how it's built. Okay, so we downloaded our data. Hopefully you were able to do the same. And you can see that this is the same Larry data set. The UMAP projection is slightly different here because we've downsampled and I reran the UMAP. I actually have a nice UMAP model that I prefer, but Colab was a little bit stingy with how to upload pickle files. So I was struggling a bit with that. So you can see we have 3 different time points in this data set and several different cell types, but you can kind of think of it as expanding out from the middle. We have this blue population, which is our initial time point from some undifferentiated hematopoietic stem cells, and gradually they make their way into these kind of terminal cell fates over the course of 6 days. And the reason I'm mentioning this kind of explicitly is because this is really useful when it comes to dynamical methods, because it's like having a ground truth for the dynamics. So in a sense, whenever we have snapshot data, which is what most people have, we never have this really real ground truth. And anybody who's in the machine learning world knows that this is kind of the gold standard for being able to develop any method. Okay, so as I mentioned, this is the link to the prescient paper. And the model that we'll create today is really based around this auto generator class that they've created. And this is kind of like where I won't really talk about my research so much today, because it's not quite as clean as theirs. It's not done yet. So I'm creating models in this kind of same area and kind of starting from where their paper leaves off. But I thought it'd be nice to highlight some of the work they did today. So this is the model that we put together. It should hopefully load. So if we just instantiate this model, we should get this torch neural network called the generator model. And here you can see that the weights, so I'm plotting the first layer of weights, they aren't initiated or trained at all. Or they are initiated, but not trained at all. So we just have this random distribution of weights. But then if we point to our checkpoint, we hopefully now have this kind of like purposefully distributed distribution of weights. Okay, so now within this data set, so I mentioned, we're going to try to look at dynamics in the sense of real time. And we won't really deal with the lineages today. So I mentioned this is a lineage trace data set, which is really what makes it beautiful. But today we'll kind of just operate on the more naive side of things in the way that we just won't pay attention to them. Because this method is actually designed for doing population level inference and then comparing to the lineages to see how correct it was. So the first thing we're going to do is we're going to sample from some day 2 cells. We're going to just create some initial sample indices to start from. And then we're going to walk forward, using this model, we're going to walk forward in the cell space from the dynamics that it's learned. And one thing I want to highlight is that the kind of beautiful part of this model is that it takes as input 50 PCs, and then it has its hidden layers. And for those of you that are familiar with PyTorch, this will be more familiar. And for those of you that are less familiar, I encourage you to check out the variational autoencoder PyTorch model tutorial from last year from this series, because I thought it was really cool. And a nice demonstration. But the key feature here is that it only outputs a single value. And the reason that it does this is because in the model, it actually formulates this as a potential value that can be then mapped back into the gene expression space. And without spending too much time kind of in the code here, we'll notice that this kind of breaks down to a drift diffusion equation that we saw in our slides. So, okay, so here this function is just going to pick a cell index for us that will get us started with some day 2 undifferentiated cells, and then we're going to grab both the UMAP and the PCA coordinates for that. And then since we have the time, the real time associated with this kind of like dynamic systems framework that we're working with, we'll note that, and then we'll take our initial cell state and we'll map this to our device along with our model. So I'll put those all on our device. And then these are just, this function here is just a convenience function. But the second function I want to show you is this Brownian motion generator, which essentially creates a random cell state that we can add to our observed cell state. So when I mentioned and I'll just skip to the end so that we know what we're doing. When I mentioned that we're going to grab some initial cell states, that would be these red dots, kind of within this cell manifold. And I know the UMAP here looks slightly different, but don't worry about that. Yours might even look a little bit different than mine since it's probabilistic. And then what we're going to use our function for, what we've trained this model for, is to learn the trajectory along these black dots here to get us all the way to the end point in these blue cell states. So, okay, so within that within this 4-day window, so going from day 2 to day 4, we're going to take 40 steps that equate to just 0.1 days in real time. And this Brownian motion generator is basically just going to generate for us a random, Gaussian distributed noise signal of 40 time steps across our 5 cells for each of our 50 PC dimensions. And then we're going to actually do the steps. So this for loop essentially says, take the current step, take this initial step and make it our current step. And then each time, loop through and pass our state through the model, and the model will take a step using our Brownian noise. And it's going to calculate the potential and map this back to our observed cell state space. And then we'll add those all together. And now this kind of 40 dimension long trajectory is kind of like our real dynamics trajectory. And then we can take that and we can plot it. So the first thing that this plotting class will do is just, it'll take the UMAP model that we've already learned and just project our PC predictions back down into the UMAP space. So that should just take a second. And then we can plot, hopefully it will look somewhat nice. So you can see where the model kind of predicts cells to kind of like stay nearby. They kind of float around and don't really go anywhere. And you can see in other cases where it really kind of pushes them along the manifold. We can try this again for other ones, and we'll see that we'll get different results for different initial starting points. So I'm going to let this run while I finish talking about the other parts here and see if we get anything that looks slightly different, just for example's sake. So now I did 15, and I'm just going to let that run the UMAP. So while I let that run, I'll talk briefly, and I know we're kind of we just have a few minutes left, and I'll leave a few minutes for questions. So we'll talk briefly about this pseudo time here. So I mentioned cell rank is a really, really useful package. It has a lot of built-in crosstalk with ScanPy, and I'm not sure if it's part of the SC verse or not, but they kind of have a lot of cross collaborators on their papers. So we can use their module, and I've included a link here to the documents that are really, really fantastic for getting started with that to get a cytotrace-based pseudo time. And here we can see I just plotted a few more lineages, and you can see some really move along this manifold space while others kind of hang out in this undifferentiated space, which makes sense. So I'll show you why I feel like this makes sense in a second. But also I'll mention that, we don't necessarily consider this model to be perfect. And in fact, that's kind of like where most of my PhD work is trying to improve these models. So let me quickly just calculate the cytotrace time here. And then what we'll do is we'll plot and I'll zoom in here. If we plot the cytotrace time along the manifold, you can kind of see what you might expect to see, where I just know from working with this data set quite a bit that these are the neutrophils here that are the most kind of developed and specified, while your more undifferentiated cells kind of hang out towards the middle here. And the reason that I mentioned that, it kind of makes sense that, in the real dynamics, we have lots of cells that kind of spin around in this undifferentiated space, even over the course of the 6 days, is that you actually do see the distribution of cells. So I've broken it here into the 3 different days and then plotted the cytotrace time as ridge plots for each. Of course, we get some population by day 6 that's kind of shifted along this cytotrace time axis, but a lot actually kind of stay in this undifferentiated state, which kind of checks out, based on what we know about these systems in a general sense. And so for the most part, I think I'll wrap up here. I could show you more things, but I think this is kind of a good introduction, and a decent place to stop. I will just quickly mention kind of a plug back in the slides. Let me skip through this stuff. So I'm part of the GETS lab and the Pinello lab. I work a lot with some great people like Anders and Roy Tong here, our graduate students and computational biologists at the Broad. I've had a lot of fun working on these things. In general, I'm really active on GitHub, and I have Twitter and all these things. So if you are interested in these types of things and want to talk more, I'd be happy to talk anytime. And feel free to reach out. I'm currently working on a package that is nearly done, and we're hoping to get the preprint out as soon as possible. Of course, as everybody is in graduate school, trying to get preprints out as soon as possible. But I would be happy to share that with anybody who's interested. And it's kind of along the same lines of these models like Prescient and some of the other dynamical systems-based models I showed you today. So if you have any questions, please let me know. Other than that, I will leave a few minutes gap here to kind of allow people to ask questions if they have them, or what have you. But I'll upload the materials or kind of polish and send them along. But yeah, thank you.
Thank you so much, Michael. That was super informative, and I'm sure I'm not the only one who thinks that they need a second to go over all the different papers that you cited and all the cool methods that are out there. Thank you so much. And if anybody has questions, please just unmute yourself or type them in the chat. We still have a few minutes left.
Oh, hi. Hi, Michael. Really good talk. Thank you so much.
Hi, Anna. Thank you.
This is going to be a really probably silly basic question, but in your predictive model, in the UMAP space, where does the gray shape, the gray background, come from? Is that just when you get data from all the cells, the space that they fit into?
Yeah, that's a great question. So here, essentially, this gray space would be all of the cells. So I'm kind of using this as a background kind of a contextual background for our predictions that we're making, where if we had sampled all of the day 2 cells, we could probably cover most of this space. But in this case, it's kind of just adding context and meant to be a backdrop for the more interesting trajectories that we predicted.
Thank you.
Yeah, great question. And glad you asked that. Seems like there's another question in the chat here. What are some of the challenges with incorporating multi-omic measurements into these state modeling? Yeah, that's a really good question. So we're kind of exploring this now. I have a collaborator who's, so I'm towards the end of my PhD. I'm trying to graduate. So a lot of things are I have a lot of questions too that I would love to work on if I had infinite time, but I need to eventually finish. So I've worked mostly on single cell attack and single cell RNA independently, but not so much on the multi-omic space. Aside from, just general analyses. When I say that, I'm more so referring to the dynamics, which is the point of your question. But yeah, hypothetically, these models are kind of agnostic to the input omic, right? So I think it would probably just be a matter of someone who's an expert in that space, whether it's protein, et cetera, applying the model to their own data, and then trying to figure out what makes sense or what adjustments need to be made in order to kind of incorporate the assumptions that are made in those spaces. And when I say that, I'm mostly thinking of pre-processing type steps. So I'm somewhat optimistic that a lot of these models are extendable towards other types of omics. And I think with velocity, probably there's a lot of papers and a lot of you'll see if you're active on Twitter, you see people kind of get in heated debates on Twitter about velocity. And I'm kind of agnostic to it all. I like to think that I am. And so I think that in the future, these models will probably get more and more informed by better understandings of how to just interpret the pre-processing level information of these data. So that's a very active space, is my understanding.
So we have 1 minute left. Oh, we're out of time. But Michael, if it would be okay with you to let people know how to best reach out, like email or anything else, I'm sure people, if needed, can follow up also. But yeah, I'll stop recording now. And thanks everybody for coming once again. And yeah, this was a great talk. Thank you so much, Michael.
Yeah, thank you for inviting me. I had a great time. And like I said, I have a Notion to go along with the slides, and I have my email and GitHub and Twitter and all that stuff in there.
Oh,
so
perfect. Great.
Yeah, so we can post that online, and then yeah, but people can feel free to, I think I'm pretty Googleable, so hopefully it's easy to find me. But yeah,
Awesome.
also you can ask Monica if you're having trouble.
Yeah, will do. All right. Thanks again.